So let's now pivot to the second PARP inhibitor trial that was also published in the same issue of the New England Journal. Incidentally, Gonzalez-Martin is the first author of PRIMA, the first author of this other study called VELIA 3005. These are both GOG trials.
GOG,
I know.
yeah, 3005.
We enroll our patients on GOG trials. Now that's not Energy. This is GOG, which is
GOG partners.
Sort of partners with pharma.
Right.
So thank you for that. So tell us about VELIA.
Well, it was an interesting trial because it looked at using veliparib in over 1,100 patients in 3 different arms. So it had a design very similar to GOG 218.
The bevacizumab study.
Correct. Which obviously has implications too, and we'll talk about that in terms of how we're using that in frontline therapy. But back to 3005 or VELIA, there were over 1,100 patients enrolled, and it was 1-to-1-to-1 randomization. And what they had was the control arm had placebo with chemo, followed by placebo, and then the 36 cycles, which worked out to about 2 years of maintenance there. And then the second arm was veliparib, a fourth PARP inhibitor in this space now, with chemotherapy. So concomitant chemotherapy followed by placebo maintenance. And then the third arm, of course, is veliparib throughout. So we call that veliparib throughout, being that it was given with chemotherapy at a lower dose, 150 milligrams BID. And then there was a run-in phase for maintenance that was done at a lower dose of 300, and then they kicked it up to full dose 400. And that went on again for approximately 24 months, 36 cycles.
We needed to do that,
3 arms.
because we needed to see if chemotherapy PARP together was a good idea. Right. And so that's why this is a 3-arm, right? Yeah. Is it a good idea?
Well, it depends on how you look at it. So again, they very much met their endpoint. The overall, the primary endpoint on this was BRCA. And it was a hierarchical design, and then it was stepped down.
BRCA's easy.
Yeah. So they hit though, 0.44. So again, impressive. And they had some differences again. And what's interesting, a lot of these studies allowed primary debulking, of course, but also neoadjuvant in all of them, which was interesting, right? So because of the dirty little secrets out now that people are using more and more neoadjuvant. You can't design a trial now with just the primary. It would take too long to enroll. But the other trials required that you had a PR or CR to get randomized. So think about the randomization time here, because people like to look at medians and so forth. The time 0 is different, right? So the enrollment, the randomization occurs up front as you start chemo, because we have to know whether you're going to get veliparib with the chemo or not in 2 of the 3 arms. So that was an important component. So your time 0 is actually in front of the chemo, so you have to add on approximately, or subtract out 4.5 months, or add on to the other trials to try to figure out. But importantly, there's preclinical data that shows synergy with PARP inhibition and DNA damaging drugs such as platinum.
That's why we had to test it.
That's why.
So is it a good idea?
That's why it had to be tested. Well, again, it hit the primary endpoint. The intent to treat, if you look at the overall patient population, all comers, with a group of patients that were a little more demanding in that they had patients that also had stable disease, and even a few those with progressive disease that were eligible for this. So there's all this argument of which patient population is the worst, but nonetheless not the easiest population, and they still hit their endpoint with a hazard ratio of 0.68. So it's pretty impressive.
So is it a good idea for the 3rd time?
Well. They were able to do so without significantly impairing the relative dose intensity. So they were able to look at that. So from a combination standpoint, it was tolerable. If we look at the safety data, if we look at the relative dose intensity, there was a small decrease, but not a significant decrease. And there was a small number of patients that benefited because those SDs became PRs, which would have made them eligible for any other PARP trial. So there's that in question. But if you look at the curves, that part of the curves, there's not a significant separation between them.
There is an increase, it wasn't emphasized, there is about a 10% improvement in response rate. And you don't need to worry about, let's say if you're a bevacizumab person, about the debulking and all of this. I had someone come up to me when I wanted someone that you would recognize, I'm not going to use their name, and he says, look, Brad.
What were the initials?
I like VELIA because I'm not going to have HRD back when I start chemotherapy. He says, look, I get it, maybe, but no, not really. So I like VELIA because I can start the PARP inhibitor, and if it works, keep it going.
That's right.
And if it doesn't work, then pivot to second-line bevacizumab. Is there traction with that message? I think there might be a little bit.
Yeah, I mean,
And it's tolerable, and it increases the response rate, and even the stable disease, you transition seamlessly into the maintenance.
yeah. No, I think it's a positive trial, as I said.
Got in the New England Journal. That's pretty good.
Very impressive, yeah. Very impressive data. And by the way, Rob Coleman, our good friend, was the PI on that
Part
trial.
of the GOG, that's right.